Thank you everyone for coming for the last seminar of the year. It's nice to see such a nice turn up in the last seminar. Today we're very happy to have with us Jackie Schiller from the Technion. Jackie doesn't need a lot of introduction, but I'm going to introduce her anyway. So Jackie is a professor of neuroscience from the Technion. She's the head of the neuroscience department there at Technion. She also been chairing ISFN for several years. Her research touches a lot of subjects that we have interest in here, and from her website or CV, said information coding and processing in central nervous system neurons, coding information in semisensory and motor systems, molecular, cellular, and network mechanisms of memory and learning, motor learning, reward system, reinforcement learning, Parkinson's disease, cortical malfunction, and autism. Yeah. And there are many papers that I can go to. That is done. I think the recent one in Science about computation in dendrites, in tough dendrites, in five neurons. Today, I think we're going to hear about the motor system. So we're very happy to have you here. Thank you for coming.
Thank you, Jonathan. I'm happy to be here. Always feels really good to come back, and especially that I'm wrapped with the cortex and pyramidal neurons of the building. It feels really good. So yeah, let's start. Okay, so my long interest in research, my laboratory is interested in how the cortex computes, learns, and stores information. Of course, these are really very generic questions that I'm sure the interest is shared by many laboratories. I hope to show you today our angle to shedding some light on these age-long questions and very complex questions. So I've been interested in how the single element in the cortical network computes information and also computes and also the plasticity of these neurons, the pyramidal neurons. And with the main question that I had in mind now for many years, how the single neuron is actually contributing to the network function. What is the load that is shared between single neuron computation and between the network computations? And most of this work was done in vitro, but with the new techniques that been ripe in the last years, I've shifted my interest and focus to study these questions in vivo, and I concentrate mainly on the motor system with a special interest on the motor cortex, and we ask questions of how we generate and learn new movement. And we study this in a cell type dependent manner. We study this in a more more sparse preparations, but also more dense preparations, where we study many neurons, but we also study single cell in a cell type dependent manner. And I hope to show you today what we've learned in this context. So why motor system? So movement is essential for life. This we all know, and it's practically the only way by which the organism is interacting with the external world. Importantly also, since our preparation is a rodent, we know that there are many similarities between the rodent motor system and the primate and human motor systems. So there are some important homologies. So this turns the mice as legitimate preparation to study questions of motor learning and motor control. So this movement is realized by a very complex system involving many cortical and subcortical regions, which all converge to the primary motor cortex, which is serving the main output controller and the main output region. So, and this is why we wanted to concentrate on the motor cortex. These cortical computations are being performed by many cell types. Broadly speaking, they are divided to pyramidal tract neurons and the PT neurons and to the intertelencephalic neurons, the IT neurons. Today I will mostly talk about our work in the pyramidal neurons, which are a specialized layer 5 pyramidal neurons which convey the information, the output commands from the cortex down to the spinal cord and to the brainstem. So by studying the pyramidal tract neurons, we actually have direct access to the functional output of the system, and this is actually a really unique opportunity to study both the input at the input level, the dendrite input level of these cells, but also see the output that they are generating. So these layer 5 pyramidal neurons were studied a lot in the past, including my lab, mostly in vitro, and we learned quite a bit about the plasticity and integrative characteristics of these neurons. Because of the interest of time, and I want to show some new stuff here, I will not give an exhaustive review about what we know about these neurons, but just some important facts that will motivate later on my talk. So the layer 5 pyramidal neuron, and I excuse if people really know this, but for the sake of students, I will give some introduction, are the largest neuron of the cortical hemisphere. Actually, in humans, they can be the size of several millimeters, 3-4 millimeters. In rodent, they are between 1.5, 2 millimeters in size, and they possess a really a big dendritic arborization. Can you see my dendritic arborization? And through this dendritic arborization, actually they receive around 10,000 of synaptic contacts from, as I said, cortical and subcortical region. They process this information. Actually, this is the question. We know a bit how they process it in vitro, and we don't know what's happening in vivo to produce the main output of the cortical hemisphere to the brainstem and spinal cord. And throughout the years, we learned that dendrites actually are not simply funneling their information to the axosomatic region to produce action potentials. We know that some complex computations may happen in dendrites. One of these is mediated by their unique properties, biophysical properties. For example, the existence of voltage-gated channels such as sodium, calcium, and NMDA receptors, which can fundamentally change the spatiotemporal integration pattern. How come? So, in contrast to the simple neurons that are used mostly in artificial neural networks, where they contain, they simply sum their inputs to generate the output, and they have one threshold nonlinearity. Actually, in real life neurons, in these cortical, pyramidal cortical neurons, location matters, okay? And I will give an example. So by location, I mean the relative location of inputs and their spatiotemporal activation pattern. So for example, in this case, if we activate a small patch of synapses, clustered synapses, we can actually, by increasing the stimulus intensity, we can actually initiate a local spike. In this case, it's a local NMDA spike, and you can see here the current voltage relationship. And this spike is actually amplifying a lot the local synaptic inputs and have impact on the axosomatic voltage. Let me give you an example where if we now use the same number of synapses, but instead of being localized to a single dendrite, now we we distribute them to multiple dendrites. In this case, actually, the computation changed from supralinear computation, as you can see here, to linear and many times also sublinear. So it's fundamentally different, and it matters where the inputs are being located. And here we can see the current voltage relationship for clustered inputs compared to dispersed input, and this is fundamentally different. Now, these spikes can come with multiple shapes and forms and ionic mechanisms. I will not go into it, but I will just mention that two very important spikes are the calcium spikes. Are the calcium spikes which are generated at the axosomatic nexus, okay, which amplify the inputs coming from tough dendrites. These are the tough dendrites, and the NMDA spikes which are generated in thin basal and tough dendrites. Now, to wrap this up, what I would like to convey is that these dendritic spikes can confer the neuron with multiple decision points and can actually also turn the neuron to a multi-compartmental neuron. So the way we now conceive the neuron being, instead of having a simple single nonlinearity, the pyramidal neuron can actually perform a cascade of complex nonlinear functions. And it was perceived as having the capacity of a small deep neural network which was shown with a theoretical work done by Idan Segev and Mickey London recently, and even before by Bartlett Mell, claiming that these neurons can be described as a two-layer neural network functions. So with this in mind, and with 25 years of work in dendrites, we have come to the conclusion that actually dendrites can provide the neuron with a large toolkit of nonlinear functions and of local plasticity mechanisms. But while this work was done mainly in vitro, we wanted to know what's happening in the in vivo conditions under relevant behavior. And since we concentrate on the motor system, under motor relevant behavior. And so basically what we are asking here is what is the input-output function of these very important neurons which are doing the computation in the motor cortex. So with the work I will describe, we will mainly concentrate on tough dendrites because they were shown to be very important for motor plasticity and motor control. So if we want to know something about the input-output function of these pyramidal neurons, we actually have to go and record from the dendrites and from the somata of these neurons, and this is what we set up to do. I would like to mention that this is really, although it is studying a single neuron at a time, but actually studying these neurons also enable us to see what remote regions are, how they exert their function of this to this neuron. So, for example, I hope to show you at the end that we can manipulate the thalamocortical inputs, and we can study in very high resolution how this is affecting dendritic function and the somatic function of these neurons. So, what are the tasks that we use? Whoa, sorry. So we use the Hendrich task, which is a central behavioral protocol for primate for a long time and also used in humans as well. And it was adapted for rodents recently, for head-fixed rodents, where we trained the mice to actually be tuned to a tone and to extend its hand grab the food pellet and bring it to its mouth. It's quite a complex movement for the hand. And the mice learned this, I would say, like between a week and 10 days. Fast mice will learn this task. We, of course, need to analyze the data, and we used softwares which are machine-based protocols. So we do both. We segment the behavior to specific features of the behavior, such as we mark when the lifts were, the grabs, and so on and so forth, as you can see here. And we also track the hand in 3D dimension using deep lab cut. And with this, we can actually know at any single moment the trajectory of the hand. We also use walking, which is a very different usage of the forepaw. And we can measure the speed, the acceleration, along with, of course, the neuronal activity. So what do we record? First of all, we wanted, as I said, to measure the activity of these neurons in a cell-type dependent manner, and this can be done in multiple methods. For example, to label the pyramidal tract neurons, what we did is actually injected retrograde virus to the pons, and actually we identify these neurons by their projection targets. And after labeling these neurons, we did 2-photon imaging through a cranial window. Chronically, we can record from these mice for many, many months. And here you have an example of such an injection. And I will show later on that we can do it in a very dense manner. So we can have a large number, many hundreds of somata, and we can do it in a very sparse manner where we have in our field of view maybe 1, 2, or 3 neurons. So this will enable us to really study the input-output function of a single neuron. We also studied, and I will show just a little bit today, layer 2-3 neurons of the motor cortex. This can be accessed by a specific injection to the layer or by using transgenic mice. So let me start by describing with a dense preparation the somata, recording from the somata, because we wanted just to know what kind of activity these pyramidal tract neurons show in mice with our behavior, with the hand-rich behavior. So here you see an example of such a session. We have here around 300 neurons. Here you can see the average activity of these neurons in a session. Down here you can see actually the distribution of the different behavioral events. And quite clearly one can see that the activity was locked to the behavioral events. Here you can see example of 3 neurons. This was very robust upon repetitions. You can see that in each repetition the behavior was quite robust, and we can see that overall the activity is movement related and follows the reaches and grabs of the hand. So to analyze, yes.
What is the x-axis seconds?
Yes.
And the color is 2 spikes or the maximum?
This is a good question. So this is delta F over F, and we don't know how many action potentials were there.
That's calcium imaging?
This is calcium imaging, yes.
So we don't know
We don't know how many action potentials. But it's relative. Yeah. It's the delta F over F signal. Okay, so then we went and studied a little bit deeper the relationship actually between motor variables and behavior, and we used generalized linear models to actually model the calcium activity and by the various behavioral variables. And here you can see that our model reliably captured the behavior. Up here are these are the real recordings the calcium transients, and down here is the model. So the model looks pretty good. And here you can see the overall relative contribution of the different predictors to the calcium signals. And what we could see is that actually the predictors, which were mainly motor predictors, constituted more than 60-70% of the total variance. So this is not surprising anybody that pyramidal tract neurons, which convey the motor program, will be motor related. However, I would like to give you an example that this is not the case in different cell types in the motor cortex. For example, this is the activity of layer 2-3 pyramidal neurons, the IT neurons, and we can see already that actually the activity is totally different. We have a three-phasic behavior with a huge inhibition, and the relationship to the motor variables is not straightforward. What we could see in these neurons, for example, a major property of a subpopulation of these neurons were their activity at the end of the trial. And what we saw is that this activity was highly correlated to if the animal succeeded in the task or failed in the task. So, for example, you can see here one neuron, and you can see this. You can see here one neuron. These are all the success repetitions, and these are the failure repetitions. And we can see that the activity is mostly starting after the termination of movement, and we can see a prolonged activity after the hand reach was over, mainly in success trials. And these are more examples of that. But some neurons, actually, they showed preferential activity to failure. So here we can see an example of, these are all the failure repetitions here. And these are all the success repetitions, these are all the failure repetitions, and we can see that this neuron was preferentially activated at the end of the trial to failure. And these are more examples.
But these can be related to motion as well, right? Because when you're succeeding, your mouth moves in a certain way, when you're failing, it doesn't. So these are also essentially...
This is a good point. And actually, we did, I will not go into it, it's a whole paper which was published with all the controls. Actually, what the conclusion that we came here is that these neurons are tuned to the outcome of the trial. So, and it's not, you cannot explain it simply just by chewing or reward or behavior. But I don't want to go into it. I just wanted to show that actually, in this region, in M1, this is M1 of the forepaw, you have a population of neurons which are not tuned simply to the trajectory of the hand or any components of the hand doing the hand reach.
But still, it's not completely, what you show is not completely true, because in some trials it could do this, in other trials, as you show here, it doesn't do it. So these are all success trials.
Yes. So it's in most of the trials, and there is probably some variability, a small variability between trials. But broadly speaking,
looks big here. Also, the timing, the onset of this, it's seconds, so it's a big variability in time, very big, much bigger than I'm used to.
This is calcium imaging, right?
I think there is a lot of, I don't know, it's not measured here, but looking at the colors, it's not, I don't know, I should say it doesn't look consistent with success in every trial, yes.
With the failure in every trial. This is the failure neurons.
So
but what the failure in each neurons. So this is the success trials up success. There. This is success and this is failure. Ken. Yeah. But what actually I just wanted to show here, just as to show that different cell types have different properties and different activity profile. So you would agree with me that this activity profile is very different from the activity profile of the pyramidal tract neurons, whatever they are coding or not coding.
Are the failure responses lateralized?
How the failure is?
Lateralized?
No. No.
But the other one, the pyramidal tract, is.
Yes.
So it's a different.
Yes.
Not the same.
So, okay. So one more point I would like to convey here, which I think is interesting with regard to these neurons in layer 2-3, is that actually these neurons actually are not there and they are developing with training. So here we can see the number of these outcome-related neurons at the beginning of the training, and when the animals were already proficient. So we can see the huge increase of these neurons. This is just another analysis that we've done. So actually what we have here is that these neurons are not innate residents of the system, but they are developing through learning. Anyway, so I want to go back to the pyramidal tract neurons, which will be the focus of my talk today. And actually, here what I'm showing is just the comparison of the GLM between layer 2-3 and layer 5 neurons at the peri-movement segment and the post-movement segment. At the post-movement segment, we actually could not model the pyramidal tract neurons. We could only model the layer 2-3 neurons, with outcome being the main predictor. And here we can see that overall the activity of pyramidal tract neurons was strongly related to movement. But actually we are left with the question how this is produced. So if we record only from the soma, what we do is asking questions about representation. We don't know mechanistically how this representation came about, and what we wanted to at the start of this project, we wanted actually to understand better how this input-output function of the neurons is being done in pyramidal tract neurons. Okay, so for that, actually what we had to do is devise a new pipeline where we will have only very few pyramidal PT neurons in our field of view. So sometimes we had only one neuron, sometimes 2 to 3 neurons, because we actually wanted to track and to reconstruct the whole tree very accurately. And whoever did tracking of dendrites, and I can see here Idan's students, know that this is very difficult. And you can be mixed up with other dendrites, so you have to have really extremely sparse preparation. So so we access these neurons through ponds as before, but then what we did is actually when we had a sparse enough preparation, we could do a Z-stack, a volumetric stack reconstruction with a structural dye, with structural red dye. And here you can see a full reconstruction. This all done in vivo, where we could really reconstruct our tree, putting a special emphasis on the tough tree. Later, actually, this is all with double labeling. We could mark our region of interest, the multiple ones, to sample tough dendrites, and we could record the activity using 2-photon microscopy, calcium imaging, and through a cranial window.
So we don't reconstruct the basal dendrite at all?
Here not. And it's actually, we are working on it now using 3-photon imaging, but with 2-photon imaging, it would be very hard to see the dendrites the way we see them with such a high resolution. I don't think one can do that. These are neurons which are 700-800 micron deep into the cortical tissue. So, and then with a special software that we wrote, we could actually assign our ROIs exactly to be mapped on the dendritic structure. Okay, and here you see an example of a reconstruction, a Z-stack that we did in vivo. You will see here 3 neurons, blue, green, and yellow. And slowly, as we go up, you can see the first, second, third bifurcation, and you can see the whole dendritic tree, which we could really reconstruct very nicely from the soma all the way up to tough dendrites. And okay, what did we see? So immediately what we saw that the activity pattern in these pyramidal tract neurons, they were all injected through the pons. We could divide it to 2 types. So one type of activity which was seen in a subtype of these pyramidal tract neurons, which we term type 1 pyramidal tract neurons. And you can see here some examples of this activity. Why don't you know? Okay, but you can get an impression. This is all one neuron. You can see that sometimes the activity is more spread and sometimes the activity is more localized. And actually what we could see is pronounced independent activity of the different dendritic regions. Here, this is one example of one such a session. And we can see that the activity spanned small regions of the tree, like in this example, like a small branch. We could see also, to our great surprise, that actually only half of the tree was activated. So these pyramidal neurons, they have a typical first bifurcation at their apical dendrites, and these are all the descendants of the first bifurcation, the green and the orange. And you can see that in this case, only the green ROIs were active, while the orange ROIs were not active. So just half of the neuron was active. This is really surprised us a lot. And of course, you can see that some of the events, the whole tree is active. This was one type. The other type, as you can see here very clearly, is that we observed a more uniform activation of the tough tree, and you can see the examples here in the traces, and we call these subtypes of pyramidal tract neurons type 2.
Jackie?
Yes.
For those of us who are not experts in them, what did you expect? So why is it surprising? What is the expectation?
So I didn't talk about it, but actually, if you look at the literature, many labs who worked in vitro shifted to work in vivo. And majority of labs saw only this type of activity, that actually the whole tree was coming off and everything was coordinated in the tough tree. But this really surprised us when we started this project. It was against all the intuition that you will have such a huge tree, and yet it will actually be activated synchronously.
That's unique to motor cortex, or because in sensory cortex you do see very sparse activation of synapses?
So I think you talk about spines,
yeah.
Right? So when you talk about spines, you talk about a small compartment, and you don't know what's happening in the dendrite, right? So nobody up to now really correlated of what's happening in these spines and the dendrite activity.
But it is possible that what Adi is saying is correct, that in sensory you have a sparse representation, and therefore you have like not entire trees.
So I would say the following, and you can see it here, Adi. So sometimes you can see these small activations here, very tiny activations, okay? So you can see some isolated, but this was not the majority. This was not the majority. The majority, I would say, 80% of the cases, you would see these spread events.
So I should say, so from a theoretical standpoint, if you have a strong activation at a proximal branch, it should it should depolarize everything that's distal to it because of the biophysics. Right, so the way you get a half of the tree light up, so you have like an NMDA spike on the proximal branch of that half of the tree. It'll light up everything distally to it.
So you mean propagation?
Yeah.
So I will talk about it.
Right.
Concerning your question, actually, so we ask ourselves, okay, so what's the difference? Okay, these are all, if you look at their somata, there is no big difference between the location of these two subtypes of pyramidal tract neurons. Their somata is actually in the same depth. So what's the difference between these two types of neurons? So what we saw actually that there is a rule. These type 1 neurons actually where we could divide our population of pyramidal tract neurons using just very simple clustering methods to two, okay? The one which actually have early bifurcation, okay? And early bifurcation and very large nexus. These actually were the type 1, which showed this functional compartmentalization. The other type, and you can see here that the somata is same size and same depth. The other ones, the type 2, were these ones with late bifurcating tuft and shorter nexus. Okay? So, and this actually we had like, I would say 100% division. Now, just looking at the morphology, I can tell you what would be the physiology of these neurons. It's very clear cut, these two subdivisions of the pyramidal tract neurons.
And the fraction? 50-50?
Oh, of the big ones? So I will come to it. Actually, the big ones are really, really interesting because these are the ones which would go to the spinal cord, directly to the spinal cord. These ones, the type 1, the more complex one. These would be the ones to go to the spinal cord. So this is the project we are doing now.
Okay. But still, out of pyramidal tract neuron layer 5, how many will be type 1 and how many will be type 2?
So I have to say that it's hard to compute from this very sparse labeling because we have like very few neurons in each brain. So from what we have, if we inject if we do the same injection to the pons, I would say like 30%, 40% would be the type 1. Not a negligible subpopulation, no.
So most of the drive is from layer 1?
Again, what's the question?
Most of the drive will be from layer 1. It's mostly in vitro. What drive those cells?
Oh, what drive these cells? Good question. Thalamus.
Directly to layer 1.
Directly to layer 1.
And you don't see
the thalamus will comprise more than 50% of the total inputs to the tuft. The other would be M2, and also they will receive from the column itself from layer 2 fluid. But thalamus will be a major driver to this tuft. Okay, so now we wanted to quantify what we could see really by eye. In the movies, we wanted to quantify. So what we did here is actually constructed activity vectors, which means we concatenated the calcium transients for each and every ROI. Okay, we have an activity vector for each and every ROI. And then what we did to see the relationship to the structure of the tuft tree, we computed pairwise Pearson correlation matrix between each ROI pair and arranged them according to the tree structure. So now we could actually talk clearly of what would be the relationship between the structure and the function of each tree. And what do we see? So here you can see an example. This is type 1 neuron with early bifurcation and very large nexus. And this is the structure of the tree. This is the structural distance matrix. You can see the ROIs that we took. We can see the hemitree of these two parts. And here you can see the Pearson correlation matrix. So you can immediately appreciate the compartmentalized representation here in this matrix. And you can see that the compartmentalization was strongly related to the structure of the tree. You can see here this hemitree very robustly. So each tree can actually function independently. For each hemitree can function independently from one another. And so this actually type of neurons mainly segregate information through their tuft branches. The other type, the type 2, here we can see again an example, and you can see that the correlation matrix was really, you could see a global correlation with no specific structure related to the structure of the tree. And actually what these neurons are doing, they are actually integrating the information, the motor information. And I will show in a moment how this activity is related to representation of motor variables. So up to now I showed you examples and analysis of the entire events. But here you can see one session from a neuron, and we observed that we had like a large variability between the calcium events. Some of the events, we could see a really long tail. So we thought that maybe if we divide the events to clusters, we could see more details when we construct the Pearson correlations. And actually here you can see clustering. We clustered the events to 4 clusters. And you can see here the very small activation, the single branch activation. These are all raw data. You can see the subtree activation, the hemitree, and the full tree. Of course, when we construct the Pearson correlation matrices, now you can see much more details, okay, of the correlations to the secondary and tertiary branches. You can see very clearly the hemitree, and of course, we could see also events which were propagated to the whole tree. Here you can see the correlation of you can see the correlation as a function of the inter-ROI distance, and you can see very nicely the correlations to the distance. This is we can see the statistics with the comparison of the type 1 in purple and type 2 in green. I will not dwell too much on that. I will just say that we used several measures to check the differences between type 1 and type 2. Here we could we wanted to measure the degree of correlation between the structural matrix and the functional matrix. So we used a measure called mantle, which is measuring the correlation between matrices. We also measured the linear regression. And also, obviously, what we did is validated all our results using permutation shuffling tests. Okay, so what are these events? And Chaim, this is a question that you ask. This is calcium. What is this calcium? What are these events? So we don't have, and in dendrites, it's very hard to actually do some verifications of what are these events. But what we can do is, and we and the community, we can actually slice these brains and do recordings, dendritic recordings and stimulations with the same sensor, with GCAM sensor, and just to see actually what do we see there. So from previous work, and especially the Arnett work, He concluded that with GCAP, actually what he could see is local spikes, and he could not see single action potentials or single EPSPs. And here we can confirm that. So what we see here is you can see recording from the nexus. This is the main bifurcation, and from the tuft. And actually, what you can see here, you can see here that the blue is the back propagation, and the blue is the calcium spike, and you can see a large calcium transient. The red is the back propagation, and the back propagation barely reaches the nexus region, and the green is the NMDA spike. But however, when you go to the tuft, we knew already, and this is confirming what we knew, that calcium do not back propagate to the tuft. Okay? Almost zero back propagation to the tuft, and you can see here actually that we could not see almost any calcium transients in the tuft. Of course, back propagating action potential do not reach the tuft, and the events are mostly signifying some amplification through the NMDA, so mostly it would be these NMDA spikes. Here we can see what's happening when we block NMDA locally around in vivo. This is done in vivo. This is the activity before NMDA blockade, and this is activity after NMDA blockade with our sensors, and this is really confirming our slice work. Okay, so what we actually see here is mainly regenerative events, signifying probably a full-blown NMDA spikes or partial NMDA spikes. Now, what is the relationship between these different segments of activity in the tree to the soma? Because this is important. Is it just locally, or can it have any significance to the function of the neuron? So when the entire tuft is active, 100% of the cases the soma was active. But also when the hemitree is active, also 100% of the cases the soma is active, meaning that this hemitree activation has functional significance, because when half of the tree is active, it can convey information to the soma. But also when a partial of a hemitree is active, we could see also activity at the soma. So overall, what we saw that the degree of somatic activation was proportional to the percent of the active ROIs at the hemitree, okay? Because the whole tree, of course, we always see activity at the soma, which means that these tuft dendrites are a strong driver to somatic activity in pyramidal tract neurons. And actually, this is not surprising because the main input of these neurons is the thalamus, and thalamus is very important in driving the whole scene at the motor cortex.
Can you say something about the delay between the two?
The delay at the soma, you mean?
Yeah, between the soma and the tuft.
Ah, okay, I see. So it's you mean who was first? Yeah. So with this resolution, this is a 10 Hertz resolution. With this resolution, we almost see them at once. It's very hard to really check a clear delay between them. But as you can see, we only rarely saw somatic activity first and then the dendritic activity. Mostly the activity would be almost simultaneous in this time resolution that
So it could be common input.
It could be?
Common input.
It could be common input, yeah. But we don't know of common input to the soma and the tuft, and also with regard to attenuation of inputs.
No, the soma is driven by synchronous activity, which is
From down, and the tuft could.
But can you distinguish that? Can you distinguish which half of the tree was activated by just looking at the soma? Or once you're at the soma, it doesn't matter? I mean, is there any information in which?
So this is a really good point. We didn't analyze. I think there is a lot of information there still to do, and also to see the activity at the soma with relation to the number of ROIs which were activated, the shape and the duration. We didn't do yet this because it has information for sure. Okay, so, but a conclusion here is that even small tuft compartment can communicate information to the soma, and this is probably due to this local amplification that is happening at the tuft, which would be mostly due to NMDA spike amplification. So, of course, this is activity. What is the functional significance of this tuft compartmentalization? Does it have any significance, or in other words, are motor variables represented in a compartmentalized manner as the activity? So here we did two types of analysis. First, I would like to show the raw data, and here you can see the motor variables for the hand reach, and this is the treadmill, and this is the velocity, and you can see that for both pyramidal tract neurons, type 1 and type 2, there is a locking of the activity to the behavioral events, as we can see here. We can see that they are strongly related, but how they are related. So first what we did is actually we extracted the peri-behavioral event times of different behaviors. For example, here, when positive acceleration, negative acceleration, or rest, and we constructed these Pearson correlation matrices only at these peri-behavioral event times. And we can see a big difference between the way, between the compartmentalization of the tuft tree to these different behavioral events. So here we can see the statistics of when we take any three behavioral events, and we can see the significant difference both in the linear regression and the Mantel statistics of the segmentation of the tree, which means that tuft is compartmentalized differently during different motor events. So it has a meaning. Actually, what we see there in the activity is the compartmentalization of the behavior in different compartments of the tree. Then we did GLM. As we did at the soma, we asked ourselves, okay, can we model well the activity in tuft dendrites? Yes, we can model it well. And we now do the relative contribution of every single ROI that we had in the tree. Here you have an example, and these were the variables that we used. And you can see that motor variables in a way, all of them are represented, but to a different proportions in the different tuft branches. And some of them are very selective, as I will show in a moment. So, oh, wow. Okay. Okay, so let me finish this. And so actually what we saw here is if we sum the GLM for the hemitrees, so you can see in this example a really cool result where onset and offset is represented in one hemitree, and the positive and negative acceleration is represented in the other hemitree. Now, if we take if I go just back in a moment, and if we take these vectors, and now we construct the Pearson correlation matrices, not for activity, but for the GLM vector, we can actually see the similarity of the vector between ROIs, and again, we can see differences between the hemitrees, and we can see the relationship to the tree. So actually, this type of neurons segregate information, and they can represent information in parallel, which means they have a capacity, and because we know that even smaller branches can communicate information to the soma. So we know actually that this tree actually has a big capacity of representation, okay? And it can represent multiple variables in a row. Here we can see that for type 2, we could not see such a segregation. And actually what we think is happening that this type of neurons just globally integrate the motor information, and maybe they can serve for signifying timing or action selection, this, which you don't need really to know what type of variables are there, while the type 1, maybe these are the interesting neurons which provide the motor plan for movement.
But type 2 could also be, type 1 could also be segregated by labels, right? They can get specific inputs for the whole tree.
So, sure, yeah.
So we don't need to be timing, we could be representing some variables that are not.
No, I mean, the type 2, which is weird, because you see that the capacity to represent different motor variables is not very big. So they just merge all the information that they have and funnel it to the soma. It's an amplified information, but they don't differentiate really at any single moment. Because I think this type of neurons, they can participate in the timing of actually providing in the dynamical system. They can actually participate even multiple times in the dynamical system, signifying the movement.
But if you take just the sum of what you see on the top, it will be like what you see on the left. You get the same thing as you see on the right.
Yes.
So overall, they seem to represent the same thing.
Yes, but the soma is actually outputting, this is at the dendrite, and this is conveyed to the soma, and the soma is actually merging everything together because all this is represented at the soma. You see, at the dendrite mixed.
So you see difference in the output between the two types in the behaviorally related soma.
So this we don't know yet. This is a really good question. I think we would like to go and see, and this is the next step that we'd like to do, actually characterize exactly, maybe I'll go there. I'll skip the simulations. So I'll go here. And so the next step for us is to understand what are these type 1 and type 2 neurons in terms of what is known about the pyramidal tract neurons. Because we know that there are multiple types. For example, those pyramidal tract neurons which avoid the thalamus and go to the spinal cord directly, and those which go to the thalamus, okay, and on their way down. And this is actually what we do here. So we inject retrogradely from multiple locations. For example, here thalamus, spinal cord. These neurons were all reconstructed from the spinal cord. And from what we know now, and we have something like 30 neurons already reconstructed, we can see that actually our reconstructed neurons are not significantly different. They are almost the same structurally as the type 1 neurons. So around 90% of the neurons which were reconstructed from the spinal cord are type 1 neurons. Okay. Now we don't have the statistics yet, but it seems that type 2 are those that send their inputs to the thalamus. So it would be now interesting to go and record the soma specifically and see what they code, what is their role in movement. So okay, I'll skip the thalamus and I'll go to the acknowledgements. So the dendrite work was done by two very talented MD PhD students at the postdoc. These are the collaborators, and this is the work I showed for the somatic recordings from the PT and from the somata.
question, and if you have more, during the happy hour.
Yeah. So I want to know what percentage of this correlation within the dendritic branching structure is just due to the biophysics, and what is due to similar inputs, right? Because from the biophysics, again, if you have a branch, you activate the branch, you should activate everything distally to it. So, of course, you'll see a correlation between a branch and its distal branches. So is that what you think is happening? That's back propagation of like an NMDA spike, or is it common inputs?
So I think it's both. So what we did this is exactly what we wanted to know when we went and did the simulations. We wanted to know what is the role of the input and what is the role of the structure and what is the role of the biophysics of the dendrites. And actually what we did is reconstructed the same, we took our reconstructed neurons and used a pseudo-random input. And what you see actually very nicely when you do a pseudo-random, maybe I'll show it to you. It will be interesting for you. Sorry. Yeah. So when you just stimulate this neuron, stimulate in a simulation with the pseudo-random input, you actually see a very nice compartmentalization of the tree, even more compartmentalized to what we see in vivo. So I think what's happening in vivo, you have more correlations, more spatial correlation, more spatial temporal correlations happening in vivo to explain, for example, that we don't see this very compartmentalized activity as we see here. So this is what you see in vivo, this is what you see in the simulation. So I think in vivo, you do have common input, which contributes to the signal that we see.
Or a bias towards more proximal inputs, right? Because if you have more inputs coming in proximally, that will create more of these events that light up the little tree.
Yeah. But I don't think it will be not proximal. Proximal. Not at the main apical, right? It should be in the top.
No, like relative to other branches. Right?
Relative,
the more practical you are on the branch structure.
I agree. Yeah. By the way, we didn't touch inhibition here at all. But this type of simulations that shows us that this is an emergent property of the tree itself. You don't need inhibition to produce this type of compartmentalization. Though possibly inhibition is there and contributing.
Okay, thank you, Jackie.
Thank you.